So in this lecture, we all will be talking about antigen presentation and processing. Antigen presentation and processing is the last topic of this unit, and thereafter we will just take a PowerPoint review of the whole unit. Now, what is antigen presentation and processing? Before that, I like to tell you we all are going to discuss how peptide fragment forms from the pathogen and their products are produced, how these peptide fragments are combined with MHC molecule, and how MHC peptide complex presented to the cell. So basically in this video, in this lecture, we all are going to discuss about these three things. So what is the process? Process is very simple to by heart. The process in which the pathogens are whatever their products are there. Not always the pathogen is harmful, right? Sometimes the product of the pathogen, like one bacteria is there and bacteria is secreting the bacterial toxin. So that toxin is also antigenic. So that could also be presented. Not necessarily always the antigen is taken inside and the part of the antigen will be presented to the T cells or the B cells for the formation of the antibodies. Sometimes pathogens products can also be immunogenic in nature, because most of the time you might have seen some insect has just injected its toxin inside your body. Insect will not enter in your body, but still you are getting itching, you are getting the immunogenic response. Why? Because the product of that insect was antigenic in nature. So their products are degraded to produce the peptide fragments, and this peptide formation of the peptide fragment is called as the antigen processing. Okay, because now here antigen could be the pathogen or it could be the pathogen product. Now these peptide fragments will be combined with the MHC molecule. It could be the MHC1 molecule or it could be the MHC2 molecule. Within the cell, this processing is occurred, and thereafter this MHC and antigen 1 complex will be formed, and this complex will be flagged on the that particular antigen presenting cell. It could be the normal nucleated cell or it could be the professional antigen presenting cell, and this cell will display this antigen to the T cell. This T cell will be the T helper cell, and presenting this particular antigen to the T cell is called as the antigen presentation. So this whole process we all are going to discuss by two ways. One pathway is the endogenous antigen, the antigen which are hiding inside the cell, and exogenous, they are not replicating inside the cell, they just move or they replicate, they increase their number outside the cell. So both the antigens are taken care and they are presented. Both the pathway we will discuss under this 3 point only. We all are going to learn first about their peptide fragment formation. Then thereafter we will be discussing combination with the MHC molecule and thereafter their presentation. So let us discuss about the endogenous antigen. Now when we talk about the endogenous antigen, that means here we are concerned with the normal nucleated cell, the antigen which goes inside the cell. Most of the time it is the virus, right? Virus, they are trying to enter in the host cell, and thereafter they just integrate their genome or they stay inside the cell. So at that time, normal nucleated cell we know forms the MHC, major histocompatibility complex 1. So they are going to form the MHC1. So what's the process? Just will give a quick review on the process. First of all, what will happen? The pathway is starting from here, bottom. Virus is entering. Now virus DNA will replicate, will form the mRNA. Now this mRNA will be transcription, translation, everything will be happen, and at the end virus will try to form its protein. Now this protein will be recognized by the cell that this protein is not the normal protein. This protein is trying to harm the cell. If the cell will get the immune response that this protein should not be reside in the cell, and anything which is in protein form and will not be in use of the cell, that is degraded by the cell organelle called as the proteosome. Name itself is suggesting it is going to work on the protein. So what is a proteosome? It is just like your mixer grinder. It chops the protein which is not of use. So it is the cytosolic organelle. That means it resides into the cytoplasm. So this proteosome will degrade this viral protein. Very fine chopping will be done, and thereafter this proteosome will form 8 to 10 amino acid peptides. Now protein is a very big peptide, so small fragments will be formed, and this peptide will be bind with the TAP protein. Now TAP is the transporter associated with antigen presentation. I repeat, TAP, it is the transporter associated with the antigen presentation. So the TAP protein are actually the reporter protein that will help to take that reporter viral protein or viral fragment inside the endoplasmic reticulum, because endoplasmic reticulum is a specialized cell organelle. It will not allow anything to enter inside. So to make the entry, they require some ID card. So this TAP protein is just like the ID which will help the viral protein entry inside that. Now why we are taking the viral protein inside? This is not of use, but here ribosome is designing the MHC molecule. Now which MHC molecule will form here? Here the MHC1 molecule will form. So this MHC1 molecule will be assembled inside the ribosome, and there this viral protein fragment, or we can say the antigen fragment, will be taken and as per its size. Now if I am stitching a dress, definitely I am going to stitch it with my measurement. So as per the designing of the viral protein fragment, the MHC molecule will be designed. That's why it has to be taken inside it, and inside the endosome only assembly of the antigen fragment and MHC1 molecule will happen, and thereafter they are will going to final processing. We know that secretory vesicles are formed. Further processing of the protein is done into the Golgi complex, and thereafter Golgi complex will take it or will transport this complex. Which complex? MHC1 and viral peptide complex on the plasma membrane through the secretory vesicle, and thereafter this complex will be flagged on the surface of the cell. So once again we'll revise the process. First, what thing is happening? Virus is entering into the nucleated cell. It can be any nucleated cell, any body cell. Then thereafter virus will, definitely virus has not just come for the time pass. It will try to make its protein. Then this protein will not be considered or will not be accepted by the host cell, because this protein is something different. So host is having the special machinery, or you can say the cell organelle, that is called as the proteosome. What is the function of this proteosome? To chop unwanted protein. So this proteosome will degrade that particular protein. Now this protein will be converted into the small peptide fragment. See, if nucleic acid fragment is more than 50 amino acid, that time it is called as the protein. But if it is less than 50 amino acid, it is called as the peptide fragment. So this peptide fragment will be very small, approximate 8 to 10 in the case of the endogenous antigen, and in the case of the exogenous it will be 10 to 12 amino acid. So small molecule that is called as the peptide fragment. 8 to 10 amino acid will be formed, and this peptide fragment will be taken inside to the rough endoplasmic reticulum. As I told you that rough endoplasmic reticulum will not allow anything to enter in. So for recognizing this particular molecule, the full form of TAP is transfer associated with the antigen presentation. Transporter associated with the antigen presentation. So this transporter associated with antigen presentation is a protein. This will be bind with this particular peptide fragment, and this will get the entry inside the rough endoplasmic reticulum. So inside the rough endoplasmic reticulum, first MHC molecule will be combined. Now MHC molecule is again a protein, so its definitely processing will occur inside the rough endoplasmic reticulum. So here the two, it is the MHC class 1 molecule consists of the two type of chain, one polymorphic heavy chain and one beta 2 microglobulin chain. So this is globular protein, and this will assemble, means it will come into its full final form. Now here inside the rough endoplasmic reticulum, this antigen and MHC1 molecule, they both will combine, and thereafter their final processing, as we know that the finishing and the final processing of the protein occurs into the Golgi body. So it will be taken to the Golgi body, and there only it will be packaged into the secretory vesicle, and from the secretory vesicle it will be taken to the plasma membrane. So this is called as the antigen processing. Now this is processing is done for the presentation. So how the presentation occur? Suppose this is the infected cell. Now see, till here we all understood that the antigen is flagged on the plasma membrane of the infected cell. Now to whom it is going to present? It is going to present to the Th1 cell. When we were learning about the cell mediated immunity, that time we had already discussed, and I told you that you have to remember MHC1, CDR8, TH1. Okay, this is the mantra you have to remember. So what happens there? How it is going to be presented? Infected cell, how it has presented antigen on the surface. I hope you all have understood. Now how it is going to present it. So suppose it has come across with the Th1 cell. See, one point you need to remember, this infected cell will not roam. It's not a wandering cell. It is a normal cell. But when we were learning about the immune cells, we have learned that these lymphocytes, they are circulated. So the thing which we need to remember, that Th1 cell will come in the circulation, and through the circulation it will encounter the infected cell which is flagging. Understood? Are you getting the point? One infected cell, suppose it is a body cell, it is a your liver cell, or it is your lung cell, whatever. So here it is infected and it is flagging the antigen on its surface. So now here this Th cell is the roaming one. Okay, this cell cannot go na, it is a fixed cell. It is a normal cell. It is a fixed cell, so that's why it will wait. Some lymphocyte should come to me, and then thereafter I will present. It happens in the small, when you all were very young in your school, you all were waiting. Teacher will come, then I will show my homework, right? So it is something like that. This infected cell will wait. It will flag, means it is just has raised the hand and will be waiting when the teacher will come and I will show my work that I have processed the antigen. So same way here is also occurring. This Th1 cell will come, and that time Th cell will feel like it will recognize. Now we have learned about the T lymphocytes, that it can recognize only and only cell which are presenting the process antigen, right? So here this TH cell has come. It will see that yes, this cell is looking unhappy. Oh, why it is unhappy? Then what it will do, it will have one special type of receptor that is called as the T cell receptor or TCR. So this TH cell will check whether this flagging part is antigen or not. Not only this, this will have the extra security, or you can say the extra type of checking. This will also have the CDR8 receptor. This will check that whatever presented antigen is showing, whether it is showing by MHC1 molecule or not. Now, why this is double checking is required? The reason behind that the TH cell can be means misbehaved sometime because they are very accurate. They are going to call, or they are going to accelerate the this T cytotoxic cell for the formation of the cytokines, and their perfect checking is must because their response is going to generate the T cytokines to produce the cytokines which are going to kill this infected cell. So normal body cells could not be harmed. Okay. So for that reason, this double checking is done. So what is going to happen? Once again, we will revise in the case of the antigen presentation. Cell has infected with the either with the pathogen or pathogen product. Okay. And thereafter, this pathogen product is flagged on the surface of that infected cell with the help of the MHC1 molecule. Okay, then thereafter this MHC1 molecule was waiting for the TH1 cell. TH1 cell has come and it will check whether the part is presented by infected cell is antigen or not. For that, it will make use of the TCR, and once if it is confirmed, yes, this thing presented is antigen, the TH cell will use the another weapon, that is the CDR8 receptor, and it will confirm yes, this is MHC1 molecule. Then thereafter it will give response, it will stimulate the T cytokines, and thereafter what happens we have already learned in the cell mediated immunity. So I hope the endogenous pathway or cytosolic pathway is clear. Now we will see the another pathway, that is the exogenous antigens. Exogenous means the antigens which are roaming outside the cell. They are not coming inside the cell. But it is not like that. If they are not entering in the cell, they are left free, go, roam, produce whatever harm you want to produce. It is not going to happen. For that, what happens? We have the different level. We have the police army, we have already discussed. Now these are the antigen presenting cells. Major antigen presenting cells are macrophages, then we have dendritic cells and B lymphocytes. I repeat, major antigen presenting cells, three are means very much professional. These are macrophages, dendritic cell, and your B cell. So best part about these cells are they are present in the circulation. They are not sitting and roaming. They will go to the junction. Now we have already learned the headquarter of the immune system is the secondary lymphoid organ. So they will personally go. They will take the effort. They will go there and they will present their antigen. So how the process occur, will give the quick review. Now see here the antigen is present outside. Okay. So now what is going to be happen? Endocytosis, we have phagocytosis, endocytosis we have already learned, right? So what will happen? This antigen will be ingested. Now ingested means they will be taken inside by the process of phagocytosis. Now we know that one phagosome, or we can say the endosome, will form. Now this endosome will be fused with the lysosome, and the lysosome have the hydrolytic enzyme. One thing we need to remember, that in the case of endogenous antigen, proteosome is helping, and here the lysosome. Okay, so this lysosome will degrade the enzyme. Now see, we have already learned the lysosomal degradation. It is independent of oxygen and dependent on oxygen. Both the pathway we have already learned. So simply what we have learned in the case of the fusion, or in the case of the endocytosis, whatever antigen is entering inside the cell, it is chopped. Now here 10 to 12 amino acid small fragments will form. Now here that fragment will not be taken outside the lysosome. It will remain inside the lysosome. Now how combination will occur then? See, endoplasmic reticulum will form the protein, then Golgi, it will process into the Golgi. Golgi will send this MHC2 molecule. This MHC2 molecule will design, and this MHC2 molecule and peptide of antigen assembly will be done inside the endolysosome. Okay, and thereafter from there only it is going to be taken outside the cell surface. Once again we'll revise the process. First, what is going to be happen? One antigen, it's roaming out very nicely. So macrophage, they will encounter that particular type of antigen, and they will ingest it. They will take it inside, and they will do the process endocytosis, and thereafter the lysosomal degradation of antigen will happen. Now antigen will be chopped into 10 to 12 amino acid small fragments, and thereafter here in the next organelle, that is your endoplasmic reticulum, the assembly and processing of MHC2 molecule will happen, and this MHC2 molecule will be taken inside the Golgi complex. There it will be properly packed inside the secretory vesicle, and this secretory vesicle will fused with the vesicle which is having the small lysosomal degraded antigen. So here their assembly inside that vesicle will happen, and this MHC2 molecule or antigen peptide will combine, and thereafter this thing will be taken on the plasma membrane, that is the flagging, and thereafter this transported material will be taken and presented to the T cells. Now here the things need to be remembered. Antigen presenting cells are professional. They are present into the circulation. So they will personally move to the TH cell. Here the TH cell will not come into the circulation. They will go. Same thing will happen. TH cell, when they will find the TH cell, they will say, hello, hello TH cell, wait, I want to show something. Then this TH cell will see, will check whatever this APC has brought. Now TH cell knows APC, because always if I am a person who is complaining again and again to the police, so police knows that this is the one who is again and again bringing the society problem, or X or Y or Z. So TH cell knows this APC, but still they are very professional. They will check whatever this APC has brought, that is antigen presenting cell has brought, is the antigen or not. So again here Th2 cell will also have the T cell receptor. It will properly check whatever thing has brought by it is antigen or not. Then thereafter another checking, this thing is MHC2 or not. So for that, it have the CDR4 receptor, and thereafter this Th2 cell will stimulate the B lymphocytes to produce or to design antibody. And how it happens, we have already learned in the humoral immune response. So this is all about antigen presentation and processing. A quick revision will do. Normal cell has infected, then after infection it has processed the antigen, and after processing it has assembled that particular antigen with MHC1 molecule, and this MHC1 molecule has combined with that antigen and flagged on the surface of the normal infected cell, and this will show that particular antigen to the Th1 cell. Then Th1 cell will check the antigen with the help of the TCR receptor. Not only this MHC molecule checking will also be done, and this checking will be done by CD8 receptor, and thereafter once it is confirmed yes this is the normal infected cell and this is the MHC1 molecule, this TH cell will take the inquiry further, and it will stimulate the T cytokine cell. And what T cytokine cell will do, it will form the cytokines, it will form the chemical which is going to destroy the infected cell. Then we have learned next is the exogenous pathway. Exogenous pathway, the main player or the main heroes are the antigen presenting cells. This APC, they will do the endocytosis of the wandering antigen into the bloodstream, and thereafter they will do the endocytosis of it. Then after endocytosis they are going to process that particular antigen, and they will assemble that antigen with the MHC2 molecule. Again they are going to flag it MHC2 and antigen complex on their surface. They will present, they will personally take efforts, they will go to the secondary lymphocyte secondary lymphoid organs, primary lymphoid organ and secondary lymphoid organ we have already learned in detail. So they will go to the secondary lymphoid organ, they will meet with the Th2 cells, they will show their presented antigen on their surface. Now Th2 cells will check whatever antigen is presented is properly processed or not with the TCR, that is the T cell receptor. Then thereafter again second level of checking whether the molecule is MHC2 is there or not with the help of CD4 receptor, and if confirmation is there done yes the presented cell is the APC and this molecule is MHC2 and they are presenting the antigen, they will stimulate the B cells. Now what is the role of the B cell? To form the humoral immune response, means to design the antibody. So this is all about antigen presentation and antigen processing. So at this note we'll stop today.